Hey, good morning, guys. I hope you are doing well. So today we are getting into a much more technical part called interpretation of sequencing chromatograms. Okay, how are we going to interpret the sequencing chromatograms? So before we start, I want you to know how it looks. I will tell you. Uh, See, the chromatograms or DNA sequencing chromatograms involves analyzing them. They are actually a machine output file. Okay, they are default machine output file generated by ABA sequences. You have to analyze a chromatogram before you pass it on to your clients. When you are working as a bioinformatician or bioinformatic research associate, it is your job to analyze the chromatogram very carefully because not always the software is right. Calling a particular base pair, the software is not always right. So manual inspection is very much essential, where the bioinformatician role is a paramount and it is a laborious task. I will show you why. Okay, so what is this interpretation? The interpretation means nothing but analyzing the peak pattern. You know, every nucleotide here is actually they have four colors for four different nucleotides. They have a peaks. Okay, these peaks are nothing but the deoxyfluorochrome molecule detected by the laser sensor of the DNA sequencer. All right. So you have to analyze the peak patterns to determine the nucleotide sequence and assess the quality of the read. Okay. I would say just by seeing this read is fine, this is fine, and here we may have to be more careful. I will tell you step by step. Before that, I want to introduce a new software today for you guys. This is okay. This software, what I have here is Chromas Pro or Chromas, you can call it. Okay. So Chromas is actually a DNA chromatogram reading software. Um, so yeah, these are the chromatogram files. These are ABI files. Okay, once you have a Chromas, it will organize as a chromatogram file. These are ABI files. These are actually machine output files. So once I load them in the Chromas, this is how the machine output file will resemble. Okay, here I have trimmed out because of low noises. I will tell you more. So I will demonstrate you how to install. This is a free program. You can do it. Just go to Google. Okay, go for Chromas. 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 So this first option. Okay, tenicilium.com.au. Go there. In this page, you will see there are two softwares here. Okay, Chromas Pro. Do not download Chromas Pro. Only download Chromas Lite. It is only just 4.4 MB. I'm downloading. Download completed. So here it is. You guys know what to do with the setup file, right? Double click it, accept the terms, okay, and finish the installation. Once you have finished, since I have already installed, I'm not installing it again. Once you have finished the installations, you will see something like this. Okay, this icon where you can open this. So this is how the Chromas Lite looks like. Today I will be emailing you three chromatogram files. You can use Chromas after installation and open those files. I will tell you the assignment at the last of this presentation. Okay. So that's how the Chromas softwares are installed and the chromatogram file could be read through this software. So what is this DNA chromatogram interpretation? Okay, the chromatograms are nothing but an automated sequencer produced 4-color chromatogram, okay, along with the nucleotide text at the top of the chromatogram. Usually, these peaks may contain, usually will contain errors, and for many reasons, I will tell you. The errors in the chromatograms are not only machine-based, they actually starts from your first process of DNA sequencing, which is DNA isolation. I will tell you how they can infer and they can cause the noise here in the chromatogram. Okay. So as a bioinformatician, we are responsible for manually reviewing the error before extracting the actual nucleotide out of the chromatograms. Okay. Usually the error may also appear at the beginning of the chromatogram, middle of the chromatogram, or at the end of the chromatogram. Okay. So we will see that one by one. I want to categorize this chromatogram inspection into 4 main steps. I've been inspecting chromatograms for more than 15 years now. I would say if you are very clear about these 4 steps, of course, will produce a high quality chromatogram data. Okay. These days, even though we have AI to do this, but still manual inspection is essential. All right. Let's see one by one. Okay. An ideal chromatogram should look like this. The first chromatogram in your left side. Okay. The peaks should be evenly spaced and show only one color at a given position. So if it is red, it is T. If it is blue, it is C. They should be evenly spaced. The space between the peaks are even, and this is normally clean. And the height of the peak from the baseline, this is a baseline. From the height of the peak is 3/4, which is a healthy chromatograms peak. And the minimal baseline noise indicates there is no baseline noise. This is baseline noise. Okay, some beats at the bottom of the baseline is a baseline noise. So a high quality sequence shows very clear, distinct peak with little background noise. This is a perfect chromatogram. But here, if you see, the baseline noise is present, but still it is okay because it is not interfering with the peak height or something. So true peaks are very clear, and this level of noise is acceptable. But when you see here, the third segment, okay, so here you may not see 221 are here, but irregularly spaced peaks are visible around 291. Okay, so here we have 290. This is a 291. See here, we cannot call this peak. We know this is guanine and guanine, but there is a cytosine peak in between these two guanines. Okay, so we need to consider this area very carefully. Okay, and also base at the position 310 is unreadable. Okay, here see, better call it as N. Okay, do you remember during our first class we spoke about IUPAC codes? Okay, so IUPAC codes, we also have N. Whenever a software couldn't call a particular position, better call it as N. But see, here the software boldly call it as a cytosine, which may not be the actual case. So how to call it as a N? Just open the okay, just open the chromatogram. When you are working with a particular file, just open the file. If I want to call this page as this is very clear peak. If say for example, here, if I, it's very much clear that there is no, see, here T is here, but these are evenly spaced A. Okay, so there is no T here. So just delete it, select the T, press delete. If you want to, if you say, for example, this is G instead of A, if you want to change it to the G, just select it, press G, or in the caps, press G. You can edit the chromatogram like this. You can change it here. Finally, file, after editing the chromatogram from the beginning to the end region. See, this is more noisier, so I just deleted all the noisy region. Okay, so after you are done with the chromatogram editing, file, copy sequence, FASTA format or plain text format. So edit, second option, edit, copy sequence, plain text. Okay, open a notepad file, just Control V, paste it. You will export the sequence out of the chromatogram file. Okay, this is done after completing the editing of the chromatogram. So we are in step one. Before you start editing, just go through the chromatogram from the beginning to the end region. See how clear the chromatogram is. You decide yourself where you are going to focus, where you are not going to focus, just skip the run. Okay, this will give you a general sense of how clean the sequence is. Step two, look for miscalled nucleotides. I would say look for evenly based nucleotide text at the top of the chromatogram. Okay, see, can you see the spacings between C, cytosine, thymine? Here, some extra space and congested area, extra space. Whenever you see such an extra spaces, just be little careful at that region. Okay, so say for example here, okay, uh, there is a slightly wider gaps between G and A. When you are looking at the peaks, it is completely fine. Okay, This G, adenine, is correctly called at 270th position here. 271st position is exactly G. There is no baseline noise. So next nucleotide is adenine, which is called at 272nd position. It is called precisely as adenine. So I would say between this 271 and 72, there is no much examination is required. Okay, but here, see the oddly spaced nucleotide N here. G here is correctly called. Adenine here is correctly called. There is no need for N. Am I right? There is no ambiguous peaks here. So there is no need to call them as a nucleotide. Sorry, any nucleotide that is N, which should be removed. So if I am me, I will open the chromatogram, select the nucleotide and remove. See why it is very important. We are going for DNA sequencing for various purposes. Some are studying mutation analysis, single nucleotide polymorphism. Some want to identify the species. Some want to study the function of the gene. I would say 18S or 16S rRNA gene sequences, if you are having 1N or 2N, that not muchly affect your result. But if you are working with a protein coding sequence, if you introduce one nucleotide, something like this, it may end up in showing a terminal codon. Your protein may be, amino acid sequence may be inappropriately terminated. Okay. You will see when you try to submit in GenBank, they will clearly see the chromatograms are not clean. You will not be accepting your sequence for publication. Okay. So protein coding sequence, this kind of skill is very much essential. I would say for bioinformatician, this is a basic skill you should have. All right. So the third category here is base calls. Okay. Sometimes they can also mistakenly interpret the gaps or noise as a real nucleotide. Okay. Here in the position 58, almost here. Okay. We have a timing at 58. There is no peak at 59. Okay. 60 is cytosine. Why there is a guanine here? Not necessary. If there is a guanine, it should be like this. See the guanine peak here in 61st position. It should be like this. Here there is definitely no guanine. So this should be carefully eliminated. That's what I told you. Better look for evenly spaced peak when you are generally checking the chromatogram at a one go. Okay. So the third step is very much useful for people who go for DNA sequencing for mutational analysis. It is very important. Is it important? Very much important. The middle picture I have shown here is actually a discovery happened during the sickle cell anemia diagnosis, where people have sent the DNA, sent this beta globulin gene. They are sequencing that to understand why the obviously changed their structure. They found out that at the sixth codon, instead of thymine, it is being substituted with adenine. That's how they know this single nucleotide substitution, single nucleotide polymorphism is actually responsible for sickle cell anemia. Instead of the protein having glutamine amino acid for their folding structure, they are having valine, which created the entire different structure that results in the disease. Okay. So what is this SNP, single nucleotide polymorphism? If you are inspecting the chromatogram, you can see here unevenly spaced, oddly spaced N. Okay, this peak, it's not a base, it's not a noise. It's very clear they both are prominent peaks, right? Either they can be thymine or cytosine. So when you are not calling, when you do not know how to call them, better call them as a N. This is a perfect example of single nucleotide polymorphism. This is not a mistake. This is not the inability of the software to call it a particular nucleotide. This is a genuine data, meaning both the fragments were present. Both the fragments of cytosine and thymine were present in the amplicon, single strand amplicon. So this is a classical case of single nucleotide polymorphism. And here also, see here in 192nd position, this is not a baseline noise. The peaks are very prominent. They can be guanine or cytosine. Okay, but see how boldly the software call it as a cytosine. So especially people who are working with single nucleotide polymorphism, they need to be inspecting the chromatogram very carefully. It doesn't mean only N in the nucleotide text is a Q for single nucleotide polymorphism. Even software will boldly call the N position as a C. So you need to be carefully checking those regions. Okay. So the first step we have seen is just scroll through the chromatogram to see how clean the chromatogram is. The second step is look for miscalls and look for heterozygous double peaks, which usually indicate single nucleotide polymorphism. So the fourth step is a very common step. Okay, the resolution of the chromatogram at former and the later stages is usually diminishing. Okay, this is because of various reasons in the technology, not with the samples. See, the ABI machine, what we have, they usually show sharp peaks in the middle region. The initial base calling and the final region, they usually be, sluggish. In the first chromatogram, I would say this is completely fine. You can consider this region. In the second chromatogram, I would say unless you can call the peak very precisely. For example, in 700 and 92nd position, there are two G's. I can simply see two peaks here. Okay, this is evident, so G is acceptable. But here, I don't see a prominent adenine peak. So calling it as two adenine region should be reconsidered. Okay, if you cannot make a base call something like this, see here, we have only two prominent peaks of adenine, but see how many adenines have been called. So when you cannot make a guess, even they can be called as G, but they are called as N. So usually you will see such a problem compounding at the later part of the chromatogram. If you cannot make a call, I would say stop it at around 800. After 800, just delete it. You cannot make a call till 800 is fine. Won't they diminish the data? That's why we go for two-way sequencing. In two-way sequencing, both the strands are compiled and we will be taking out the 1500 base per region out of it. Okay, so that's why two-way sequencing. Even if you are deleting the bases after 800, anyway, we will compile it with the forward sequences. We will recover that by complementation. I will also produce one more video for how to compile the chromatograms, but since I feel this is too much for you, these four steps are enough to get a clean chromatogram. Okay. When you have a chromatogram, even at the initial stages, something like around 300 and 330, see the peaks, I would say the sequencing reaction failed. Okay. Why a sequencing reaction fails? Both if the laser or the capillary, there can be many problems with the machine. Also, people who prepare the DNA. See, in microbiology, I see many students are struggling to make a last quadrant isolated colony. They do not know how to do the quadrant sticking. Okay, basic microbiology technique, you should be able to produce isolated colony, which is a proof that it is a pure culture. Pure culture means single species in a plate. Many times Bacillus subtilis and cereus, they form a translucent colony around, lactobacillus and many more bacteria. They live, they coexist. Okay. So if you pick such a colonies and go for DNA isolation, you will get a DNA, you will get a sharp band in PCR only after sequencing by observing the noisy chromatogram. This is simply because of 2-3 species. The 6-ness amplicons of 2-3 species actually forming a background noise. Okay, this is a very clear-cut indication that it is a mixed culture. Why? Because the peak height, they are all more than nearly 50% of the chromatogram height, right? So if you have more degraded DNA, if you have more DNTTPs uncleaned in your PCR amplicons, you may have a background noise, baseline noise. Okay, the noises should be here. When you have a chromatograms like this with background noises, it is very much sure that this is a mixed culture. Now the problem starts. Bioinformaticians say that it is a mixed culture. When you tell this to your client, you have given us a mixed culture, they will not be accepted. There will be many things. You have already invested your investment into producing this chromatogram. If re-sequencing, you need to again charge them. It will complicate the business. That's why I feel these barcode biotechnology guys are smart. They accept only pure cultures for sequencing. Okay, they take live cultures, they themselves do the DNA isolation, PCR, they prepare both forward and reverse for sequencing, and they get the sequencing. Okay, so that's how. So to sum up, I want you guys to see this classification published by these authors. This is an optimal peak. Okay, the peak A. Peak B is still acceptable. If you have a multiple peak like this from a single position, not acceptable. This is a asymmetric peak. Misspaced peak, you should be very careful about the nucleotide text called at that peak position. Okay. Here we have a heterozygous peak in the diploid. Okay. These are typical SNPs. Misleading heterozygous, look for the calls at the top of the nucleotide text. We have shutter peaks, smiling peaks, downstream peaks, or upstream peaks. These are all diminishing peaks. I would say rather don't try to fix those kind of peaks. Just delete them and go ahead. This is a typical peak where you should call it as N. Okay. So these are the ways you have to edit your chromatogram. So this software. Okay. So how are you doing today? Again, I will tell you one more demonstration when you are watching this. Go to Google, type Chromas, the first option, click it, go here. There will be two options, Chromas Pro. You are supposed to download Chromas 2.66. Okay, just click it. 4.4 MB file will be downloaded. Go to the exe file, double click it, follow the instruction, give the permission and everything. It will get installed. Once installed, I will send you the files today. So I will send you three files. Okay, you are supposed to identify which file you are going to, which file is a workable file, and other two files have either degraded DNA sequences or they may have a mixed culture sequences. So ignore two files and select the file which has a very clean sequences. Extract it. How to extract the DNA sequence? So I would say, see here, maybe after 40th base pair I will consider. Remaining this I don't want to work, even though if you can spend some time you can work it. So I'm just deleting this. That's it. From 40th base pair I have a 10th base pair here, 20th here. When this is done, edit, copy sequences, plain text. Okay, open your notepad file. Okay, maybe open a notepad, plain notepad, paste a sequence. Okay, maybe I will name it as my sequence. Okay, my sequence. This is a FASTA file format, right? Whenever you place a greater than symbol, then whatever you are writing after that is considered as an English text. The hard enter will tell the software that it is a nucleotide sequence. So below this, you paste all the sequences you are going to take from your blast analysis, find the species, take the reference sequences, go for phylogenetic tree, and then the pairwise Excel sheet data. Okay. So this is what we are going to do today. At the end of the day, I will circulate you the Google form to get the data from. Okay. So if you have any questions, you can always send me a message. I'm always available. Okay. Is there anything else we need to discuss? Do email me or message. Thank you.